Hi Year 12s, tonight we're going to talk about experimental design in reference to the enzyme prac that we've done in class. So first of all, it's really important that you have an understanding of these four terms because they're going to come up quite a bit in this course, in particular in Unit 3. So when we talk about experiments, there are a number of variables that influence the results. First of all, the independent variable is the one that we're altering during an experiment. So there should only be, in a very well-controlled experiment, one independent variable. And obviously by altering the independent variable, that is going to have an effect on the results. The dependent variable is the variable that we're actually measuring. So by changing the independent variable, that will have an effect on the dependent variable. If we think about a prac that we could design in terms of looking at the effect of temperature on enzyme activity, then the independent variable in that case would be temperature, and the dependent variable would be the rate of activity of that particular enzyme. Also, in an experiment, it's really good to have a control. A control allows us to compare the results to check that the independent variable is having an effect on the dependent variable. It's also good to check that the experiment is working. We're going to see in a moment that sometimes it's not always possible to have a control, but in most experiments, if you can have a control, it's great. And then there's another term that pops up a little bit about a well-controlled experiment. A well-controlled experiment is one where all other variables except for the independent variable are kept constant so that they have no effect on the dependent variable. So the thing we were talking about before with temperature and the effect on enzyme activity, if we wanted to check that it's just temperature having an effect on enzyme activity, we would have to keep every other variable constant that would have an effect, such as pH or amount of substrate. So this has just been taken straight from VCAR. You'll notice if you've been looking at Unit 3 exams that they really love to have questions on experimental design, and they're usually worth 4 or 5 marks, so it's a good idea to have a bit of practice at doing it. So what they expect in their answers is one, that you've included an experimental group. So that's the group that you apply the independent variable to. So in the case of the temperature versus enzyme activity, you're altering temperature, right? And then a control group where the IV is not applied if possible. With the enzyme prac that we've done in class, it wasn't possible to have a control because you can't control the actual pH of a solution. You couldn't take that out. So it's not always possible, but if you can have a control, it's great. Now with those two groups, so the experimental group and control group, the only thing that should alter is that the experimental group has the IV applied, the control group doesn't, but every other variable that may have an effect on a dependent variable needs to be kept constant. So that's what I was talking before about with having pH controlled, or the amount of substrate controlled, or the amount of time that you measure it for controlled, right? So you're trying to ensure that no other variable is having an effect on the thing that you're measuring. Also, VCAR expects that for an experiment to be perfect, to be as accurate as possible, you need to repeat the experiment many times or make sure that you include a very, very large sample size. So I always say to you guys that, to ensure that you're satisfying that criteria, say that you're repeating it 1,000 times. It sounds quite odd, but if you're doing it in a lab, you're probably going to do that anyway. Now, a hypothesis, we haven't really talked about this year yet, but from previous years you would know it's an educated guess. And in biology, we like to use a particular convention for writing the hypothesis, and it's in the form of if the independent variable ... then the dependent variable ... So the example I've given in terms of temperature would be if the temperature is increased, so that's the IV there, temperature, then the rate of enzyme activity, so that's a dependent variable, will also increase until an optimum temperature is reached. So it's just a simple statement, it's easy to test, and it's easy to verify whether your hypothesis was correct or not at the end of the experiment. We don't require big paragraphs, just the one sentence, and make sure that you've mentioned the independent variable and the dependent variable. So let's have a go at designing an experiment. This has been taken from a VCAR exam. So it's talking about a hormone. So vitamin D is found in high levels in some tissues. So a scientist has predicted that vitamin D may play a role in the development of arthritis, and hence treatment with vitamin D may reduce the development of the disease. So this scientist wanted to test this idea by using a strain of laboratory mice that normally develop this arthritis. So what you need to do is design an experiment to test the prediction made by the scientists. You need to include 1, a hypothesis stating what you are testing, it's 1 mark. 2, you need to outline the experimental procedures that you follow, 3 marks. And 3, describe what results would support the hypothesis that the scientists made. So I'm going to give you about 5 minutes to do that. So you ready? All right, in the meantime, I'm going to watch these beautiful cows do a dance. Like I don't care, baby, by the way. Because I'm happy. Come along if you feel like a room without a roof. Because I'm happy. Come along if you feel like happiness is the truth. Because I'm happy. Come along if you know what happiness is to you. Because I'm happy. Clap along if you feel like that's what you want to do. Here come bad news, talking this and that. Well, give me all you got, don't hold it back. Well, I should probably warn you, I'll be just fine. No offense to you, don't waste your time. Here's why. Because I'm happy. Come along if you feel like a room without a roof. Because I'm happy. Come along if you feel like happiness is the truth. Because I'm happy. Come along if you know what happiness is to you. Because I'm happy. Clap along if you feel like that's what you want to do. Bring me down, can't none bring me down. The love is too high. Bring me down, can't none bring me down. I said, bring me down, can't none bring me down. So, hopefully you had a go at doing that question. So this is the answer. It's really important that you read every part of that question. So the first part was that you had to write a hypothesis. So what they've said here is the treatment with vitamin D, so that's the independent variable, reduces the chance of mice developing rheumatoid arthritis. So that's a dependent variable. So in this case, they've listed both. It is okay to see to start at saying that treatment rather than if the mice are treated with vitamin D, then the chances of developing rheumatoid arthritis are reduced. Either is fine. The experimental design, you remember it was 3 marks. So they're looking for 3 things. First of all, they're looking for that you're using 2 large groups because we always talk about large sample size, increasing accuracy of the experiment. So they've said about 20 mice. So 2 large groups of similar, so I said in class, you can even say genetically identical mice, which normally develop rheumatoid arthritis. So obviously those 2 groups, one is going to be the experimental group, and they're going to be treated with vitamin D. The other group, the control group, are given a placebo and do not receive vitamin D. Now, if you haven't seen this term placebo before, they do it a lot with human drug trials, and basically it means that you're given a tablet but you don't know which one you're receiving. Now the third thing that they're looking for is that you're controlling any other variable that might have an effect on these mice developing the disease. So factors such as diet, space, water, and temperature need to be kept constant. So the examiners need to see that you're at least trying to, account for these variables by keeping them constant. You don't have to mention all of them because obviously would be quite a few, but just so they can see you're trying to do it, then that's fine. The last point of this question, so the last mark, was something that a lot of students, I think this is in 2008, forgot to do, and that was basically saying what results would support the hypothesis. So the scientists predicted that being treated with vitamin D would reduce their development of arthritis. So for that to be correct or supported, fewer mice that are given vitamin D should develop the disease than those in the control group. So see how we're using the control group as a comparison. How do we go? Hopefully you went okay. If not, you need to come and see me. We can go through it. But there's just one more thing I need to talk about, and that's graphing results. So we talked already about the independent variable and the dependent variable. So every experiment will have both of those. When you're graphing these results, so if you had some sort of quantitative data that you're graphing, you need to ensure that the independent variable, which is time in this case, is put on the x-axis, so on the bottom, and the dependent variable, so the one that you measured, is put on the y-axis. Now, depending on what kind of data you have, if there's time on the bottom axis, you do a line graph, but sometimes it might be more appropriate to do a bar graph, especially if you've only tested, certain temperatures, but not a continuum, if you know what I mean. So yeah, line graph here is appropriate, and it shows you the relationship between the effect of the independent variable on the dependent variable. Always make sure that you label the axes, because a lot of students forget to do that and lose marks there. And also including, which they haven't done here, but including the units, so time, that could have been in minutes, height could have been in meters, so ensuring that you've labeled those correctly, and having a title as well. So for this particular graph, you could say the effect of time on height, right? So you're mentioning the independent variable and the dependent variable. So thanks guys very much for listening to my little podcast. I hope it gave you a little bit more insight into how to design an experiment. We'll be talking a bit more about this in class. Thanks guys.